Bio students, this will serve as a brief introduction / reminder of some of the things that we've already learned about DNA and will help prepare you better for your experience at the Science Museum in London. So now that you've watched the entire video of Race for the Double Helix and you have a good sense of the history of that struggle to discover the structure of DNA, we're going to look a little bit more at some of the detail about the structural components just to make sure that you can identify some pieces in the model. So you might remember at one point when Watson and Crick were realizing that it was really about the direction of the strands. Crick realized that based on photo 51, which is actually what we see over here underneath Rosalind Franklin, Crick realized that the strands should actually be running in opposite directions. And that's called anti-parallel. That word wasn't used in the film, but we're going to use it when you get back from London. So you're going to look for evidence in the model when you're there for the strands running in opposite directions. So what that means is one strand is sort of running up right at the same time while the other strand is running down, right, to create the helix. So what you're going to do is look for the sugar. Look for the sugar, the pentose sugar component of each nucleotide, and try to see what direction it's pointing. And if you can identify that, you should be able to have evidence for the anti-parallel nature of the two strands. Okay. You're also going to try to look for the different pieces and identify where the hydrogen bonds are holding the strands together, where a nucleotide would be found within the entire model, that sort of thing. Okay, so you're going to try to kind of analyze the model, break it down, and identify pieces. Okay. Here we have a little bit more information about DNA structure. So just remember, DNA can exist in two forms: chromatin form, which is the more typical disorganized form, and then every once in a while the DNA will wrap up into chromosome form, but that's only for cell division, so that's not the time that, or the way that we usually expect to see it. Here's a nice sort of simple schematic for what DNA looks like. So here we see the two strands. The dashed lines that are evident in between are representing the hydrogen bonds that connect complementary base pairs. So we see that on any one strand, there's always a single phosphate group, a pentose sugar, and a nitrogen base making up an individual nucleotide, and then we bond nucleotide to nucleotide along a strand to create a big polymer. But the important thing again is that the strands are complementary, so that if we know the sequence of bases along one strand, like this image, we can see there's a G followed by a T, then a C, then an A, we can predict what's going to be found on the other side, because we know that wherever there's a G on one strand, it will always bond with a C on the other strand. And that's what's really meant by the complementary base pairing nature. If there were a different base here other than a G, the hydrogen bonding positions wouldn't line up accordingly, and so you wouldn't be able to hold the two strands of the molecule together. Okay? And this reminds us again, adenine, thymine, cytosine, guanine, right? We're going to talk more about that, but you might notice that some of them only have one ring in their structure, whereas others have two rings. And so no matter what way we look at it though, since an A is always bonding with a T and a C with a G, when you create the bond across the two strands, you're always going to have three rings making up the sort of width of the DNA ladder. Because again, there's always a one-ringed pyrimidine bonding with a two-ringed purine. We haven't learned those terms yet, but we'll talk more about them when you return. And then this shows you how the hydrogen bonds, the atoms are positioned accordingly between those bases so that the hydrogen bonds can hold the two strands in position. Okay? Okay, so now you're seeing a bit of the worksheet that you're going to be completing while you're in London, so please remember to have this printed out and take it with you on the trip. You're going to see some instruction about what to do with the model. So here you're just going to record some of your observations, comparing it with the film and the images that you saw there. Then you're going to look at the structure a little bit more, and you're going to identify the backbone of the molecule and try to figure out what parts of the nucleotide make up the backbone. Then you're going to look again for that anti-parallel structure that we talked about before, right, showing that the strands are running in opposite directions. And then you're trying to figure out, how is the code really stored within each strand? Where is the code? What part of the nucleotide really stores the code? And try to make sense of that. And then there's a couple of questions for you to take a look at and identify as well. Now, what you may want to do before you even go, this is good practice, you can also do this while you're there with your group, is you complete this chart to make sure that you're using the right terms. So describe the structure of DNA from a big molecular level, okay, meaning how many strands is it made up of, that sort of thing. What shape is it? Okay. Function, so just what's the purpose, right? We know it's about storing the genetic code. Okay. The monomer name you should be familiar with at this point, and actually is right below this in one of the images. The 3 parts making up a monomer, and then what the examples are, what the actual names that we use to describe each of the 4 monomer types. So if you fill that out, you should be halfway there in terms of identifying the molecule and its structural features. Okay. And then finally, after you're done exploring the DNA model, then you're going to move into a permanent exhibit called Who Am I? That's all about human genetics. Students normally really enjoy this exhibit because it's very interactive and it talks all about what makes you you. So you're going to find one display of interest. So spend some time exploring first. Find one thing that really speaks to you. Take a photo of it, okay, or maybe a short video clip, but make sure you've got a nice visual. And then you're going to relate it to as many themes of biology as possible, and all of the themes are listed down here below. You can use some of this space below to just take some notes, but your key thing there is to take a photo of the image and to relate it to themes, and we'll talk about how to use that, what to do with it when you return from London. All right, best of luck, have a great time, and I will see you upon your return.